Welcome to the Deep Sea Soirée. My name is Carol Preston, and I'm with Greater Farallones National Marine Sanctuary, and will be one of your hosts tonight, along with Cordell Bank National Marine Sanctuary and Greater Farallones Association. It is really good to be here with you tonight for what promises to be a really fun and fascinating evening. We're so excited to share some newly explored deep sea habitats from around the National Marine Sanctuary system with you. And then I did want to just acknowledge that for some of us, it's been a really tough 19 months. So I thought it might be interesting for you to know that the stars of our program tonight, the sponges, have been helping us out through this pandemic. An antiviral derived from sea sponges was approved as a treatment for COVID-19 by the U.S. Food and Drug Administration in May of 2020. Species like sponges and some corals are soft-bodied and don't move much, so leaving them really open to predation. In response, they have developed strong toxins and poisons to keep predators away, making them remarkably potent medicines. And our speaker tonight, our featured speaker, Dr. Amanda Kahn, actually specializes her research in sponges, and she will give a talk on the secret lives of sponges, understanding ancient animals at their own pace. But before we start the talk, we did want to take you around the National Marine Sanctuary system. And these are your National Marine Sanctuaries, and there are 15 of them and 2 National Marine Monuments. And so we can't really dive in all of the sanctuaries tonight, but we'll start in Texas at Flower Garden Banks. And you can tell just from the name that that's going to be a beautiful spot. And we'll take a dive and hopefully see some coral, maybe even spawning. And then we'll go over to the Northwest Hawaiian Islands to Papahanaumokuakea National Marine Monument. It's a monument. And this is actually our largest marine protected area under the U.S. Flag. And it is actually bigger than all of our national parks combined. And if you really want to know how to pronounce the name of that National Marine Monument, we'll put a little chat and a link that pronounces it correctly, unlike I probably just did. And then you might notice that there's 4 National Marine Sanctuaries right off the coast of California. And that's because the California current drives one of the most productive ecosystems in the world, actually, right here off the California coast. So we're going to take a dive down into Monterey Bay looking for some octopus. And then we'll go to our 2 host sanctuaries of tonight, Cordell Bank National Marine Sanctuary, where the research coordinator will take us on a dive and show us what she's been doing. And then we'll also explore various places in Greater Farallones National Marine Sanctuary. Both these sanctuaries are just off the coast, a little north of San Francisco, and they both expanded in 2015 and doubled in size. So we really have over 50% of our sanctuary to explore, especially in the deeper areas. And we'll be sharing that with you tonight. This is our program that we have planned ahead, exploring all of our sanctuaries and diving in them, and then having a mini break with Tyler of the Greater Farallones Association, who will teach us how to make some sponge diver cocktails. And for those of you who pre-registered for tonight, you did get a recipe, and I hope you were able to find all the ingredients because these are going to be really fun. And for those of you who didn't get to register, we will put the recipe in the chat box, and I guess you'll have to improvise for the ingredients. And I know we've all just come off of Halloween and the Day of the Dead, so we're kind of keeping that spooky theme, and thought we'd take you down to meet some of the creatures of the deep, and hopefully it won't give you nightmares. And then we'll do some more sanctuary dives. And next up will be a really fun interactive program where you get to vote on the best dressed on the red carpet anemone. And you will do that through the chat box. And our speaker will start at around 7:30, a little bit past, and she will talk about the secret lives of sponges.
Cordell Bank National Marine Sanctuary was expanded to more than double in size. And these are areas that nobody has ever seen before, nobody has ever explored. It's a very productive area of the ocean. There's a high concentration of seabirds and marine mammals that come there to feed. There's also seafloor habitat that likely includes some sensitive habitats and species. A healthy ocean ecosystem provides so many services to us: food, jobs, transportation, carbon sequestration. And to protect the ocean, we need to understand what's there. And right now, there's about half of our sanctuary that we've never even looked at. And that's what this mission is all about, to go to these new areas that we've never explored before. And by doing this, we'll be able to understand the organisms, the habitat that's in our sanctuary, and to be able to better manage and protect it for the American people.
Aloha, ekomamai everyone. Welcome to my home adventure bar here in Pacifica. My name is Tyler Mears and I'm a marine science instructor with Greater Fairlawns Association and the National Marine Sanctuary. In soirees past, maybe you have enjoyed some of my cocktails. For this year's Deep Sea Soirée, we thought we would do a little bit more of a hands-on workshop. And if you registered for our soiree, you got a nice little cocktail card with the recipes. But if you haven't, we'll definitely put that in the chat so you can work on the recipe later. We're going to start things off with my personal riff on a Don the Beachcomber's classic. That is the Pearl Diver's Punch, or the Pearl Diver. Don first started it in about 1937, first started crafting this drink, and come the 1950s, a lot of his bartenders started to streamline the drink, but I'm a bigger fan of the 1937 classic. So starting my riff on this drink, usually calls for 1.5 ounces of Puerto Rican gold rum, but I wanted to bring things a little closer to our deep sea sponge communities. The closest one I could find to an awesome sponge community in Johnston's Atoll in the middle of the Pacific was Koloa rum in Kauai. So I've decided to use 1.5 ounces of Koloa Reserved in its place. Next, it calls for 3/4 ounce of Demerara rum. These are from the Demerara River, and you could use a couple different options here. I actually decided to blend two together. I went with a blend of Eldorado 5 and Hamilton Ministry of Rum Collection. Now, Don would prefer a little bit more aged, maybe an 8-year Eldorado or an 86 of the Hamilton, but use what you have on hand. You could really use any kind of a Demerara. I also subbed out the 1/2 ounce of Jamaican rum for Rum Rincon. This is from Bonaire in the Dutch Caribbean, and it's made with yerba hole, which is a nice odorous basil that this rum is made with. And there's a really great sponge community in Klein Bonaire right off the coast there. So trying to bring Atlantic and Pacific sponge communities together in this drink. We got 1 ounce of orange juice, 3/4 ounce of lime, and Don's famous gardenia mix. Now, you can totally make this at home. I was not that brave this evening. I bought some. BG Reynolds is a great way to go. But you can use, it's got butter, cinnamon syrup, honey, vanilla, lots of really great flavors, and you get a nice kind of gardenia-like odor from the pimento dram there. Nice allspice kind of scent to it. The drink usually calls for a teaspoon of falernum, but keeping it on theme with our sponge communities and keeping it in the Caribbean and Bonaire, I decided to use the native Bonaire Kadushi. This is a liqueur that's made from limes and cactus and adds a different element to the drink. And then you're going to blend this in a blender for 15 seconds. They're very specific about this. No more, no less, 15 seconds. And then once you have that all blended up, you put it through a medium strainer into a pearl diver glass. Now, these pearl diver glasses were lost to time for about 40 years. Even with the advent of eBay and collectors putting things out there in the world, they were hard to track down. But Jeff the Beach Bum Berry out of latitude 29 and his friends at Cocktail Kingdom managed to track some down, and now we have them today. So very characteristic, very classic glass. And you can see I've already whipped up the drink here, and it's all ready to go. But we got to garnish it. We got to garnish it. And I was thinking, well, how do we garnish something appropriately for a sponge diver cocktail? How do we garnish something appropriately for the soiree? And so I thought we had to kind of kick things up a notch. And thinking about sponges, sponges do something pretty unique, pretty different. They have a lot of toxins that they put out to help protect themselves from predators and parasites. But we can't put toxins into a drink. So how do we kind of bring this sponginess and this kind of tingling toxins together? Well, there's something called a Szechuan flower or a buzz button flower. It's also known as the toothache plant. And when you put this into a tincture, when you infuse it with something, it gives you this kind of tingling sensation on your lips and on your tongue. So that's great and all, but this is the sponge soiree. This is our deep sea soiree. And so I have to turn this into something a little bit more spongy. So I've started off with some flavored vodka in a nice big bowl with some of this infused spilanthes or buzz button tincture. You can order it online. You can make your own, entirely up to you. And the recipe for all this is in our notes, but it's about 7 ounces of your favorite flavored vodka. Recipe typically calls for peach. I use pineapple. And then about 2 good dashes of this spilanthes or buzz button tincture. Now, to get this to thicken up into a nice foam, we're going to use soy lectin. You could totally use meringue powder, but I feel like results may vary. So we're going to go with a ½ teaspoon of this soy lecithin, never quite known the right way to pronounce that. And we're going to dump that in. Give that a good swish around there. And now I've seen this done with a whipper, like an icy whipper canister that's charged. I've seen it done with an air stone from a fish tank, which might be appropriate for us tonight. Even just your average whipping mixer for baking. But I like to use this little milk frother for coffee. It seems to work with the batch size that I'm doing here. And the key here is I'm using a really large bowl, and I'm making sure that the foam has enough room to propagate. And this gets a really nice frothy, this might be splashing my camera here, but really nice frothy consistency. And it's gonna whip this foam up for us. And it starts to get very spongy. And you can see it's growing as I kind of continue to let it froth here. And what we're gonna do is we're gonna spoon this off onto the top of our drink. And that might be enough. That might be enough. But I wanna take things a little bit further. There's a really amazing things that used in confectionery. It's called edible luster dust, and I have some bright silvery luster dust here. This will give us our foam a nice glass sponge look to it. So I'm going to drop that in, and we're going to give this one final little whip before we get ready to spoon it off onto the top of our drink. And this will have a very nice sparkly deep sea sheen to it. And I think we're just about ready to go. So give this a few spoons into our drink here. And some of you might be saying, Tyler, all this molecular gastronomy stuff, crazy buzz button tingling foam is a bit much. No worries, we got another cocktail coming to you. But now that I have a nice, nearly overflowing head of foam, I can top it with a nice purple orchid that I was very proud of myself I got to rebloom. And there you go, you have the sponge diver. Cheers. Ooh, you can really get those notes of Demerara and that Kadushi cactus lime liqueur just coming in slightly at the end there. But simpler cocktail here for you. This is the Nautilus. This is from Victor Bergeron. He put it in his 1972 Trader Vic's bartender book, and we are going to recreate it here pretty much in the exact same spirit. So you'll want to start off with a Reposado tequila, 2 ounces. I like to use Fortaleza. I found this is really good. I'm much more of a rum person than I am a tequila, but I really like the way this tastes. Cranberry juice, the freshest you can get. Lime juice, fresh squeeze. All these citruses that I've mentioned, always do fresh squeeze when you can. And then ½ ounce of sugar syrup. This is something pretty simple to make at home. 1 part sugar, 1 part water. Boil it up, make a nice syrup, have it cool in the fridge. I really would recommend a nice Demerara syrup. It's really rich tasting, adds a nice different element to it. And then you can give it a shake and serve it up in a nice Nautilus glass. But we're not done yet. We're not done yet. You could give this a simple little orange twist of a garnish. You could do cranberries rolled in chamoy and tahini. Might be a good way to go. You can also do a simple little mint sprig here. But if you do this, it's really nice if you give it a smack. Maybe you've seen bartenders do this. Really helps burst those vacuoles, express all those nice scents. You can put that in there. But that might be enough. But this is the deep sea soiree. So I have one more little trick for you guys, and we're gonna see if this works. But I've soaked croutons in lemon extract, and this gives them a wonderful ability to hold a flame. And so we're cooking with gas. We do have a little bit of fire. Always be careful. Safety first, of course. But we're going to see if we can light this drink. Here we go. Ooh, and it's holding a steady little flame here, which may or may not show up on camera. But this is the Nautilus, something very easy to whip up for our deep sea soiree tonight. I hope everyone enjoys. Love to answer any questions afterwards. And thank you all so much for joining our deep sea soiree. Enjoy some spooky or maybe not so spooky creatures here.
Okay,
yes.
This
looks like a baleen whale, and you can still see the baby.
Yes, the baleen is still there. Oh, this is great.
5, 6, 7, 8, 9, 10. Covered in scavengers. It's a happy day to be an octopus.
Oh, that octopus looks like it's taking a little taste or sniff with its suckers. Can you zoom on those worms, please?
So those are the bone worms?
Look at
Oh, that's not bacterial, Matt.
No, they are worms.
That
Wow.
What is all that?
What is all that?
Is that larvae being... No, wait, larvae don't. What is happening?
Got the ribs. You can see the spinal column.
That is still soft tissue.
Crazy.
Yeah.
There is definitely was something.
So those might be the
Yeah,
ones
approximately.
Oh, this is so cool.
I think that is them.
meters, 450 meters.
Hope you're enjoying the soiree. My lips are still tingling from that buzz button foam on the sponge diver, and the Nautilus is still lit. Cheers.
Right.
There we go.
Oh, hey, big fella.
Is this enough octopus for you, Joe? Oh, wow.
I can see it ripple.
That's great.
That whole mantle there.
Wow.
Oh, this is great.
Wow.
Oh, there we go.
Oh my
that's
gosh,
so
she's
nice.
posing.
That's
This is great.
so
Yeah,
pretty.
he's a show off.
Oh man.
All right, we've only got another 30 seconds or so, unfortunately.
That's
Okay.
okay. I'll take every
Understandable.
second I can
Yeah,
get
we'll
with
still
this
take
guy.
it.
Oh, yep, she has eggs
Oh.
facing outwards with tentacles extended. But the majority that we've been seeing in this area are in what looks to be a brooding pose.
everyone, vertebrates and invertebrates, land lubbers and deep divers. Welcome to the 49th annual Soirée Red Carpet Anemone Spectacular. My name is Hercules DeVille, and I'm joined by our carpet correspondent, the well-known designer and decorator crab, Pierre Kelleped. Say hello, Pierre. I agree. I can feel the luminescence tonight. I'm also joined by my friend Argus Octo Armani here in the booth. Argus, it's so great to spend another Soirée Red Carpet event with you.
Well, Herc, we've had a long run of bringing ocean science and art to the world, but never before have we pushed deep sea fashion to the forefront. I know red light doesn't penetrate all the way to the deep ocean, but you wouldn't know it tonight. That red carpet anemone doesn't have a tentacle out of place.
It sure doesn't, Argus. What's that, Pierre? Our first guest just arrived, and it looks like we're starting things off with a bang. Firework Jelly is on the red carpet. Argus, who is Jelly wearing tonight?
Jelly's wearing an explosive gown designed by the East Socoran designer Halatrephes Massai from the well-known design house Revelagigetto Archipelago. A little more transparent this year. Firework Jelly revealing a little more mesoglea than we've seen in the past.
I love it. It's very celebratory. But I don't know if they've truly realized the niche of the red carpet anemone.
Ouch! Wow! Harsh, Pierre. That stings. So much for going first. I, for one, thought Firework Jelly had flair.
Well, keeping things gelatinous and venomous, the siphonophores are on parade down the red carpet anemone.
That's right, Pierre. Each siphonophore is one big, gooey, Gucci family of fashion-forward organisms. Some only pump water, some only pull in food, but they have to stick together to pull off this signature look.
I just don't see the fashion in it. Signature, perhaps, but sweeping in, it looks like a feather duster.
There's some crinoids and worms that might take offense to that remark, Herc. I think siphonophores are magnificent. The family that drifts together stays together, and they're drifting right off the carpet.
Thanks, Pierre. Slowly working their way down the red carpet is bubblegum coral. They're famous for perfect posture and striking a pose for the anglerfish paparazzi. Argus, what is bubblegum coral wearing tonight?
Bold choice today, Herc. Bubblegum is wearing the up-and-coming designer Paragorgia tonight, and they look paragorgeous.
Always accessorizing, Bubblegum is famous for the perfectly placed brittle star, necklace, or even an unconventional red crab fascinator.
I'm sorry, Herc. Although I appreciate that they serve as a breeding ground, shelter, and feeding space for a wide variety of species, that peachy color does not do it for me this season. It's just the pits.
Thanks, Pierre. Clad in iron and bedecked by sponges, the USS Independence is scuttling down the red carpet, and they aren't sharing the carpet with anyone. What a presence.
We're witnessing history here, Herc. The Navy intentionally sank the USS Independence in 1951 near San Francisco. So this organic look is 70 years in the making.
I think it's absolutely marvelous. Take a bow, USS Independence.
Oh, Pierre, what is the calamity on the red carpet? It's mayhem down there.
Terrible tentacles, Argus. This is the cephalopod seal of the century. The squid, Planktoteuthis, has mimicked the siphonophore family's signature look.
Oh, this is awkward. That siphonophore silhouette is unmistakable to predators and prey alike. I don't know, Herc, who wore it best?
Planktoteuthis has elevated the original design. Those shining iridophores give the mantle sparkle, texture, and structure. It really pops.
Well, we'll have to talk more about this audacious fashion scandal later, because holy holoplankton, it's giant larvation on the carpet.
Giant larvation designed and secreted this bustle dress themselves.
And by secreted, we're talking about one of the most versatile and sustainable materials of the season, miraculous mucus. Snot couture has gone hot couture.
Serving both form and function, giant larvation can use this boogery ball gown to filter feed and ensnare their prey. It's simply captivating. This is stunning. A few years ago, we had a blanket octopus make our fashion faux pas list when they crawled the carpet wielding Portuguese man-o-war tentacles at predators and paparazzi alike. What a contrast. The shimmering and shifting satin ensemble we're seeing tonight, Argus, is incredible. I simply must know the designer.
That would be the multi-talented and multi-armed designer Tremoctopus violaceus. Frankly, Herc, I find the detachable train a little gimmicky, although I see the practicality if you're about to be eaten. But our next star is the one I've been waiting to see all night.
Is that Deepstaria enigmata?
Those green ROV 10-centimeter lights can't look away, and who can blame them? Deepstaria is diaphanous, mysterious, gelatinous.
I'm so tired of this trend of amorphousness this award season. We much prefer the regality of the blanket octopus.
Pierre, we're arguing between a glorified Snuggie and a shower cap? That hurts, Pierre. That really hurts.
That's right, Pierre. It looks like our final guest tonight is harp sponge wearing Chondrocladia lyra.
It's a revelation! Extraordinary! That will be on the cover of the next issue of Submerged Stone magazine.
They are squeezing every ounce of elegance out of this whimsical design, maximizing surface area for feeding without a single spicule out of place. Chondrocladia hails from the Peripheral School of Design, and they've been evolving their look for 640 million years. Pinnacle of fashion.
Wow. Well, I think that's all we have time for tonight, Herc and Pierre. We've gone from Dior to Siphonophore, from Shimada to Prada, and now it's time to head inside for the premiere of The Secret Lives of Sponges.
Fish and fashion fans, now is the time to cast your vote in the chat for tonight's best dress. Thank you for joining us on the Red Carpet Anemone. This has been Argus Octo Armani, Hercules DeVille, and Pierre Kelleped signing out. Enjoy the rest of the show.
Hey everyone, so nice to see you again. I hope you're having as much fun as I am and learning as much as I am tonight. It is with great pleasure that I get to introduce our speaker, Dr. Amanda Kahn. She's a researcher and assistant professor with Moss Landing Marine Labs and San Jose State University. Dr. Kahn's research focuses on one group of suspension feeders that is highly successful at surviving in the deep sea, the sponges. There's much we don't know about these often overlooked animals, yet they form the foundation of whole unique communities throughout the world's lakes and oceans, including vast sponge gardens and sponge reefs in the deep ocean. Dr. Kahn's research has involved studies of deep sea sponges from extinct underwater volcanoes off the coast of California, the unique glass sponge reefs of Western Canada, and the cheese-bottom sponge grounds in the fjords of Norway. She received her master's in marine science through Moss Landing Marine Laboratories via California State University, Monterey Bay, and her PhD in ecology from the University of Alberta in Canada. Please join me in welcoming Dr. Amanda Kahn.
you so much for inviting me to speak tonight. I'm so excited to get to talk to you about the portion of the deep sea that I study, which is the life that lives on the deep sea floor. And specifically, my research aims to explore the secret lives of sponges, what they contribute to their ecosystems, and the timescales that they operate on, with a special focus on the deep sea. So let's jump right in. First, let's start where we are. We're here on land. This is what many of us think of when we think at first of the ocean. I'm speaking to you from Monterey Bay in California, and this view is just south of us along the Big Sur coast. We have these sheer cliffs with pounding surf, and this nearshore coastline, while not a very hospitable environment for us, is a great habitat for a variety of animals that can live just below the water. And even further down beneath the water's surface, we see a wealth of ocean resources in our sanctuaries. So here you see beautiful kelp forests that, combined with the microscopic phytoplankton, are responsible for taking up to 26% of the carbon dioxide that's released from fossil fuels that are burned and human activity. So hugely important to our climate and to their ecosystems. They also support dense communities of invertebrate and fish species, including many edible species. Those species in turn support larger animals like seals and whales. And so we have these amazingly productive, diverse regions in our nearshore environment. And I'm here to talk about how that same environment extends beyond our coastline into the deep sea. So here, this is Moss Landing Marine Labs, situated in the heart of Monterey Bay, which you see here. And we're sitting at a pretty unique spot there because just offshore in the Monterey Bay National Marine Sanctuary is the Monterey Submarine Canyon. So this canyon, if you were to drain Monterey Bay, would rival the Grand Canyon in depth and in just sheer slope. But it's a little hard to access the deep sea, so to be able to explore the submarine canyon without draining the oceans, we can't just dive. This is about the diving limits for a recreational scuba diver. So we need technology. So all of the research that I'm going to talk about here was made possible with technological advances that were done by groups like the Monterey Bay Aquarium Research Institute, and like the footage that you saw from Nautilus and from Sanctuary Cruises. So this is a remotely operated vehicle, and it acts as our eyes and our hands underwater. So you can see this is the Monterey Bay Aquarium Research Institute's ROV Doc Ricketts. You can see its name there. It has high definition cameras that allow us a close-up view of the seafloor, and it has robotic manipulator arms that allow us to actually physically grab samples or to bring instruments down to study the life that lives down there. We have a variety of collection drawers and types of instruments that can be plugged into this. So it really is an amazing platform to be able to send down into the deep sea while we're safely up on the ship, and to go and explore these habitats. And what do we see when we go down into the deep sea? We've already seen a wealth of it from the videos from before, from the Cordell Bank and Farallon Islands National Marine Sanctuaries, but I'll show you a few more. So we see all kinds of really bizarre life living in this twilight zone. And what I mean by twilight zone is that actually not a lot of light reaches the deep sea. And so you end up with these adaptations for animals to be able to live in an area where there's very low light. This is the barrel-eyed spookfish. It actually has a transparent head, and these are not its eyes. Its eyes are actually these green orbs here inside of that transparent head, and it uses those huge eyes to be able to gather the small amounts of light that can possibly get to it from those depths. So the deep sea is dark. There's also not a lot of food in the deep sea. So there are many adaptations that animals have to deal with living in an area where food is quite scarce. This is an anglerfish, and this anglerfish has a lure that you can see here at the front. That lure can have bioluminescence or light that it emits that can help to attract prey toward its mouth. Anyone saw Finding Nemo, you probably remember that they almost got into some trouble there with that anglerfish. It also had, this particular species also has many of these sort of sensory projections that help it to feel and detect any prey that might be swimming past it in the deep, dark water. And those adaptations to the deep sea are also things that we see on the deep sea floor where I study. So the deep sea in general is a tough place to live in. The water is very cold. It's near freezing, about 1 to 2 degrees Celsius. It's completely dark. There's not very much oxygen and there's very little food. So any food that gets down there, like this scrap of fish here, is a treasure for animals that live on the seafloor, like this deep sea crab. You also get interesting adaptations where food limitation really helps to drive evolution. This is a deep sea tunicate or sea squirt, and most tunicates are filter feeders, but in the deep sea, this particular group has also gained the ability to become carnivorous. So it actually looks kind of like a giant mouth, and it functions a little like a giant mouth. So if a crustacean swims inside of this gaping hole, then it can close up that mouth and digest its prey. And here, these are hydrothermal vent worms who have amazing associations for being able to eat poisonous, sulfurous compounds from the water and actually metabolize them in partnership with bacteria. It's an amazing adaptation. Why would that come about? It's because food limitation was a key driver for what these animals needed to be able to survive in their environments. Now, most of the deep sea floor looks something like this. This is the Abyssal Plain, and this particular location is at a deep sea long-term study site off the coast of Point Conception called Station M. And you can see all these critters running around. Now, this is sped up. They're much slower moving, like you see here in the display with this sea pig. This was footage taken from a time-lapse camera that took a photo every hour. And with that, you can see many sea cucumbers zipping around, snuffling in the sediments. Back there, that nose-shaped guy is a sea urchin, also snuffling around in the sediments. And as you keep on watching, you'll notice that there are globs of darker brown material that shows up on the seafloor. So we see that there are these pulses of food that come from the surface of the ocean. And then as they sink as marine snow, they feed the animals living thousands of meters below. But only 1 to 3% of the food energy that begins at the surface actually reaches the deep sea floor. So despite seeing so much life down here and so much diversity, they are all driven to survive these low food environments. And also in this view, you can see several different species of sponges. I'll point out 3. There's this guy, this little hot dog shaped sponge. There's a little volcano shaped sponge over here, and this tulip shaped guy right here. So we also see quite a few sponges even in this sparse environment on the deep sea floor. There are also areas of the deep sea that look quite different. So this is a hydrothermal vent habitat. It's a spectacular environment. These can be quite short-lived, likely only 1 to 100 years. And what happens here is water seeps into the sediments and then becomes superheated by geological processes occurring in the mantle of the planet. And then that high heat leaches metals into the water, and then that water billows out of these chimneys and vents. Here you see a black plume. There can be big chimneys that form from it, and you saw these massive worm populations with symbiotic bacteria that are allowing them to metabolize some of the sulfurous compounds that come out of there. And these support thriving communities. But those heavy metals that are leached include copper, gold, silver, manganese, and many of these are metals that are becoming limited in above ground mines. And so deep sea mining is a process that's being explored to harvest from these deep habitats. And so these huge communities, we need to understand the deep sea. We need to understand what's going on down there because humans are actually becoming more and more interested in reaching it and able to access it. So we need to understand the life that lives there and what protections are needed to sustain those communities. So those are some of the habitats that we see. And now I want to switch gears to my particular critters of interest and the topic of tonight, sponges. So starting out, what do you think of when you think of a sponge? I'm guessing the first picture some of you might have is a kitchen sponge, or you might think of sponge cake. SpongeBob SquarePants is a good one. Well, in the ocean, sponges are animals, and they are quite diverse. They're found throughout the oceans in all kinds of habitats that have water, from lakes and rivers, to wetlands, to tide pools, and down to the deep ocean. These pictured here are all sponges in tropical coral reefs, and you can see how many there are and how big they can get. This one is as large as the scuba diver next to it. They can come in all sorts of colors, shapes, and sizes. These sometimes also have associations with symbionts that affects the ways that they feed. But I'm interested in these guys for what they eat. So sponges are efficient filter feeders, which means they eat by pumping huge volumes of water, up to 900 times their body volume each day, through a filter to catch particles from that water. So here you see on the left a green fluorescent dye that was used to show water flowing out of that sponge. It's a non-toxic dye. And water travels into pores on the outside surface of the sponge and then flows past these feeding chambers where there are cells that both pump the water using flagella. You can see those here in the scanning electron microscope image. And these cells are also involved in capturing the food. They can catch the food with up to 95% efficiency. And then water and waste are ejected out of the osculum of the sponge, this sort of chimney or vent, this big opening. Now sponges eat bacteria, which are very, very tiny, but which make up 10 to 30% of the organic matter that's produced in the ocean, and which are too small for most other animals to eat. And in shallow habitats, they can change the water quite a lot. Here is a sponge that I suspended in some seawater for 30 minutes with microscopic microplastic beads. And just 30 minutes later, it had completely cleared the water of all of those tiny particles. So they're extremely efficient filters in the water, and because they process so much water, they have potential to really affect their environment. Other people are also interested in sponges. So humans have interacted with sponges for thousands of years, beginning back thousands of years ago with Greek fishermen. So here you see a Greek sponge diver who is diving down using a heavy stone called a scandalopetra. He collects a sponge on the seafloor, and then he tugs a rope and is pulled back up to the surface to bring that sponge to the surface. So the sponge diver industry was a really big deal up until about 100 years ago. So they would bring the sponges up, they would look something like this, and after some cleaning, this looks pretty familiar. This looks like a sponge. Like I said, this was a huge industry, and so sponge divers were responsible for a lot of the sponges people used up until about 100 years ago. Nowadays, the kitchen sponge that you use is likely made in factories out of cellulose. Although you still can find natural sea sponges for sale as cosmetics applicators and in painting supplies. Sponges are also major sources of pharmaceutical compounds. About 30% of the marine-derived compounds that we use for medical purposes come from sponges. This deep sea sponge here on the left, Latrunculia austini, was recently found to produce a compound that was effective at killing human pancreatic cancer cells. So there are many medical treatments that sponge extracts have been valuable for to humans. And finally, humans have been interested in recent times in actually copying from sponges and learning from them. So sponges make their skeletons out of glass, silica, which is extracted from the seawater. Now, those silica rods that the sponges produce as part of their skeleton can transmit light in the same ways that fiber optic cables can. But sponges do this just using seawater, and they produce these flexible glass rods that work great. We need to use high temperatures and heavy chemicals for doing the same sorts of processes. So there's a lot of exploration in learning how to learn from how sponges produce their skeletons. And just speaking about skeletons now, I'm going to shift now back to the deep sea, and there the sponges have pretty hefty skeletons that make up the reason why we call them glass sponges. So sponges that live in the deep sea are primarily from the class Hexactinellida, and hexactinellid sponges make their skeletons out of glass. And here, this is one of those glass sponges from the deep sea, and you can see the skeleton here is just a whole bunch of these beautiful glass rods. Some of them are labeled with a fluorescent label here, so you can see their shapes more clearly. And 80 to 90% of the weight of a hexactinellid sponge is the glass of the skeleton. So we call these glass sponges, and I'm probably going to be calling them glass sponges throughout the talk, so I wanted to make sure that I identified them that way. So I use technology to study deep sea sponges. Most recently, before starting at MLML, I was a postdoc in Jim Barry's benthic ecology lab at the Monterey Bay Aquarium Research Institute, and we went out using remotely operated vehicles from MBARI like the Dock Ricketts and like ROV Ventana to study the sponges that live in the sanctuaries around our coastline. And I use technology that's then hooked up to that remotely operated vehicle to take measurements from sponges down on the deep sea floor. So here, this is an oxygen sensor, just as one example, so that we can measure the respiration rates of sponges down in the deep ocean and understand their energy budgets, which we'll talk about near the end. We can also use it to collect samples so that we can understand what species exist in those deep sea environments. So my research focuses on two topics, and I'm going to talk about those two topics a little bit today. So the first is, what effect do deep sponges have on their environment, and how do they do it? And I think these are really important for understanding our deep sea communities and how they work. Now, the best place to ask these questions is where there are a lot of sponges that we can find and where they live very densely in an area. So what effects do sponges have on the water column and on the seafloor around them? And what are the energetic needs? What do they need to be able to survive? Now check this place out. This is up in Western Canada, off the coast of British Columbia, and they form these dense reefs. And when I call them reefs, I mean reefs, same as corals. A reef is defined as an area where individuals of later generations settle and grow on the past generations that came before them. And you can see here the skeletons of past generations of sponges and more sponges that are growing on top. And you get these really dense habitats. So I moved up to Canada and I studied these glass sponge reefs with my PhD advisor, Sally Lees, at the University of Alberta. And we studied the effects of these sponges on the habitat around them. We also traveled further north and studied the glass sponge reefs in northern British Columbia, where the two species that form in the southern reefs, Heterocone calyx and Afrocallistes vastus, are also joined by this large bushy sponge that looks to me like a pile of potato chips, Pharea oca. Interestingly, these same species of sponges are found throughout the deep ocean, but for some reason they only form reefs in a few choice spots. And for these species, the only spots that they've been found are in British Columbia and the very southern part of Alaska. So we don't know why they show up there, but they do, and they show up in very dense numbers, even though you can find these individual species growing elsewhere in a much sparser way. Now, this is not the only area of the world where we get these dense sponge grounds. So I've also traveled to study sponges in the Atlantic. So in the Atlantic during my postdoc, I moved to study the oyster beds in the North Atlantic. So this is another dense sponge community, and I wanted to study these sponges to compare them to the ones that we studied in the sponge reefs. And so in green here is the modeled area where it's expected that those species show up, and we studied them off of Norway, where they look something like this. So all of these white mounds here are different sponge species that form large aggregations that Norwegian fishermen call oyster, which translates to cheese bottoms. And that's because on sonar, these sponges would look like a bunch of holes in Swiss cheese. And then most recently, we've studied here in the Monterey Bay National Marine Sanctuary the beautiful, diverse coral and sponge grounds that tend to form. So this is Sur Ridge. It's about 30 kilometers due west from the Rocky Point restaurant in Big Sur, and it's well within the Monterey Bay National Marine Sanctuary. And you can see the sponges here. These sponges are 1 meter tall and 1 meter across. Whew. They're huge, and their large size means that these guys are going to filter a lot of water. And so we're studying these sponges in this dense habitat to again say, okay, why is this happening? Why are we getting these dense, beautiful aggregations of sponges, and how are they affecting the habitat and the communities that grow up around them? Now, I've talked a little bit about sponges as filter feeders. I want to talk in general about filter feeders. Why do I care about them so much? Well, filter feeders are important for moving food energy from the water column to the sea floor. In general, by being filters, filter feeders will concentrate particles that are too small to be captured individually, and they'll concentrate them from huge volumes of water. They can be pelagic, meaning that they drift in the water column like these sea butterflies, pyrosomes, or which we call sea pickles, or salps, or they can be benthic, living on the seafloor, like the sponges that you see here and there, or like bivalves, clams, barnacles, sea squirts, or even small protists. And actually, the largest filter feeders are also the largest animals that we have. So baleen whales could be considered filter feeders because they have baleen to capture krill and concentrate it from large volumes of water on their baleen plates. Now, the effect of filter feeding is quite important to the deep sea because remember I said the main food supply for the deep sea is food that rains down from above. So animals that can capture food from the water column and bring it to the deep sea floor, whether because they are on the deep sea floor or whether because, well, they poop and that goes to the deep sea floor, or they die and their biomass sinks, that can have big effects on the water. And it can have big effects on the communities that can be supported. And so through my research, I've focused on sponges, and I'm going to show you just a little bit of the insides of a sponge to get an idea of how these guys work. So this is a glass sponge. This is the one from the glass sponge reefs. And if we look inside using a scanning electron microscope, I could see two things. First, these are the feeding structures of the sponge. You can see the flagella that I mentioned that pump water. Two things I want to point out from these views. One is that the glass sponge tissue is really sparse and stringy. There's not a lot of tissue in there. So remember I said there are a lot of glass. 80 to 90% of their mass is glass. And there's just not a lot of tissue to them. So by not having a lot of tissue, they don't need to eat a lot because there is less of them to feed. The other thing that I want to show here is that I saw that sponges could capture particles as small as 100 nanometers. These are small microplastic beads that I fed to the sponge, and you can see they are being captured and engulfed there. And these are small bacteria that I fed to the sponge and was able to trace to see that those were captured as well. And to give a scale of how small 100 nanometers is, the width of one of your hairs is about 100,000 nanometers. So those are tiny, tiny particles. Also, by studying the feeding of these sponges, I wanted to know not just what do they capture from the water, but what happens to it after they capture that food. And so we found sponge poop. Why is this exciting? Well, the spheres that you see in here are the orange particles that you see from up above. And the sponge had created these fecal pellets that were up to 1,000 times larger than the individual particles that they had eaten. Now remember I said sponges are eating bacteria that are typically too small for other animals to eat. But by concentrating them into these larger fecal pellets, this means that they're big enough to be able to be eaten by other animals on the deep sea floor, like this hungry sea cucumber. So it may sound gross, but actually a lot of animals eat things that were already eaten by other animals. Where food is scarce, recycling is super important. And it was really eye-opening to study how important sponges were in the sponge reefs for taking a food source that other animals couldn't, meaning bacteria, and they've made it into something as fecal pellets that other animals theoretically could eat. Now, when we then went to study Geodia, the sponge in the North Atlantic, I didn't get the same idea that sponges were skinny and didn't have a lot of tissue to feed. And so here, this is, we went out in Norway and we collected sponges and we studied their feeding in the lab. And you can see here, this is a cross-section of a Geodia sponge. And if we zoom in with some of the microscopy that we did, you can see that this is no skinny sponge. It is chock full of material, and that material actually is not a lot of sponge tissue. In fact, all of this is bacteria that are living inside the tissues of the sponge. You do still see some sponge cells here. That's actually another sponge cell. But mostly we see just how densely packed this sponge is with bacteria that are living inside of it. And so because that sponge is full of bacteria, it actually feeds in more diverse ways. So we did see bacteria that were captured by sponge cells, so they were still filter feeding, but we also saw that in other areas of the sponge's tissue, sponge cells were actually roaming around and eating the bacteria that lived inside the sponge tissues. In essence, it seems like this species has figured out a way to get food by farming. And the bacteria that live inside of the sponge likely take up dissolved sugars that are in the water, which again, most animals cannot directly take up. And still other sponges have found even more ways to capture food in this food-limited deep sea environment. So every sponge you see here has gone a radical direction of switching from being filter feeders to becoming predators. So all of these sponges have given up their filter feeding ways and now catch microscopic crustaceans from the water. So what's the takeaway from this? Well, I think it's that not all sponge species make their living in the same way, and all have adapted to survive in the deep sea where there's little food available. And not just survive, but in some places thrive enough to live in dense communities like sponge reefs. To get at the question further of what sponges need to be able to survive, especially given their important ecological role in their environments, we're now studying the energy balance of a sponge. Again, using amazing technology. This is Deep PIV, which is developed by Kakani Katija and her research group at the Monterey Bay Aquarium Research Institute. And we're using it to study the flow of water through sponges. So here, this is a sponge with a sheet of laser light across it and a camera viewing that laser sheet. What you get is a side-on view like this. And once we have it oriented correctly, we can turn off the lights and we can see with that camera in the sheet of laser light the particles that are drifting and coming out of the sponge's osculum, or that chimney. And from that, we can measure the pumping rates. So far, it looks like they pump at about 1 centimeter per second, which aligns with what we've calculated using other methods in the past. And so with this, we can really see and model the flow and the amount of water that's coming out of sponges and how that flow interacts and how it travels once it exits the sponge. So when you see a sponge sitting on the seafloor, you may first know that they look like lumps sitting on a rock, but actually they are working very hard pumping water through them and filtering out bacteria. And by studying the pumping using various forms of technology, we've really gotten to go out and study this phenomenon in greater and greater detail. So this was on a recent cruise to Sur Ridge, where we used IRIS, which is another piece of technology from Kani Khatija's research group, to study the three-dimensional flow coming out of a sponge osculum to get those pumping rates. When we get those pumping rates, we can start to ask, what is the energy balance of a sponge? First, by getting the pumping rates, we understand the amount of effort that the sponges are putting into pumping water and filtering that water. And if we measure the oxygen that's consumed while doing an activity, then the energy, that energy can be measured using oxygen. And so just like we would calculate the cost of different activities by measuring the oxygen used, like with this runner here, we can do the same on sponges. So here is a sponge that we're measuring oxygen consumption from, and we can then study how much oxygen the sponge is removing from the surrounding water. And we find so far that it's not a lot. They don't take a lot of oxygen to be down in the deep sea, which means they're not spending a lot of calories. And because we know from human nutrition, the amount of calories out needs to balance with the amount of calories that come in. So if sponges are not using a lot of energy or don't need a lot of energy to pump and feed, then they don't need to be in an environment where there is a lot of energy available to them. So in a way, these sponges are able to survive in a food-poor environment by being fairly sparse themselves. The one exception to that is that Atlantic sponge, which has all of those bacteria who are also involved in getting it a lot of food, and that guy uses a lot more energy. We're still figuring that one out. So going back to our questions, what effect do sponges have in their deep sea environments? Well, through finding diverse food sources and then excreting larger particles, they act as oases in food-poor habitats. And the nutrient recycling and the transformation of microbes into usable food for other animals is a really important mechanism for survival in the deep sea, and it's driven by sponges. This recycling is so key. And how do sponges do this? Well, we found a lot of different strategies. So they must either adopt a low-cost lifestyle so that they don't need to eat much, or by finding novel ways to catch food, such as that bacteria farming or by becoming carnivores. Now, the final things I want to leave with is that sponges generally seem to work on their own timescales. So remember this time lapse from earlier in the talk? Well, I want you to watch it again, remembering that every photo was an hour, but this time I want you to watch the things that don't move, the sponges. Let's focus on this tulip-shaped one here. And that sponge, which looks like a tulip, is about to bloom and then contract. So, oh my gosh, sponges contract in the deep sea. What are they doing? Why do they have these behaviors? Well, that contraction cycle takes about 2 weeks to happen. So it's a very slow process. And if we only visit there and get a snapshot, we might not see these animals that are operating at much different timescales. Science takes a lot of hard work, but it's also really rewarding. By just being attentive and being observant, we can stumble upon things we didn't even know to look for, like these contracting sponges. And I want to end today by just showing 2 more recent discoveries that we had in our sanctuaries just by being out and stumbling upon things we didn't know. So this one you saw a little earlier. So this is the Octopus Garden. This is out at Davidson Seamount, which is part of the Monterey Bay National Marine Sanctuary. I was out on the cruise when they found these octopuses because we were going to Davidson Seamount, which is known for its coral and sponge habitats. These octopuses were not on our radar. We didn't know that they were there, and we stumbled upon them. So this is where discovery and exploration are so, so important, and why our sanctuaries are amazing places for protecting habitats that, it turns out, have things like this, where we have these brooding octopuses by the thousands that are taking care of their eggs. And on that same cruise, we ventured further south to the Channel Islands National Marine Sanctuary, where we stumbled upon and discovered the first ever glass sponge reef off the coast of California. So remember I said they were only known from up north in Canada and Alaska, but now we've seen near the Channel Islands a glass sponge reef that's beautifully dense. And we're not sure which species these are yet. We're still in the process of describing these. This is brand new info, but it's pretty exciting that these are within our coastal sanctuaries. And I think that these reef-forming species like the Phareas are really special when we come across them. And there was recently a new species of Pharea that was discovered off of Cordell Bank and the Farallones. So this is Pharea cordelli, named after the Cordell Bank National Marine Sanctuary. So it's pretty exciting how much we have in our sanctuaries. So I'm going to stop talking now because we should chat. I want to end by acknowledging the many, many people that I've worked with. Science is not a solo endeavor, and especially deep sea science is not at all possible to do alone. So I've worked with many people from the facilities that you see listed here, including key collaborators from the sanctuaries. And I thank and acknowledge all of them. And with that, I am happy to take any questions, and I also welcome questions if you'd like to email them to me later as well. Thank you so much.
Thank you so much, Dr. Kahn, for that deep dive into the world of sponges. I learned a lot from that awesome talk. Thank you so much. My name is Jean Alape, and I'm from the Greater Farallones Association, and I'm joined back by Carol Preston from the Greater Farallones and Cordell Bank National Marine Sanctuaries, and we're here to moderate this awesome live Q&A. We've got some great questions already coming into the chat. Please keep them coming. We've got some time for questions, and as Dr. Kahn had mentioned, if we run out of time, we can definitely email them to her, and she'll get back to you. So the first question that kind of came up, Tyler asks, what factors affect the distribution of these deep sponges, if that's known? Is it things like current, complexity, any other factors?
Oh, yeah, that's a great question. And it is somewhat known. We have ideas. And then also there are a lot of times when our research is basically based on why are they where they are? You know, why do we find sponges where they are? But definitely because they are filter feeders, having currents is important. So water flow is a big factor. And also depending on the species, the substrate type really matters. So many species rely on a hard surface, like a rock substrate to attach to. And then in the sponge reefs, they need to have those past skeletons of those earlier generations that they can settle and grow on. Oh, Carol, we can't hear you. Yeah.
Thank you. I've had lots of technical problems this evening, but I'm glad most of you don't know that. So now you do. Hey, thank you. That was such a great talk. I really loved it. And just thanks for sharing all of your work. And there's a great question from Barbara, and she asks, are the sponges attached to the sea floor?
Oh, good question. Yes, sponges are typically attached to the surfaces that they're sitting on. So they'll use their spicules, their skeletal pieces, to help anchor against the rocks. They also, if they live in soft sediments, can project those spicules down into the sediments, almost like anchors. Interestingly, though, not all sponges stay attached. So one of the things that we noticed from the time-lapse camera footage was that hot dog-shaped sponge, occasionally we would see those become detached from their anchoring spicules, and they would roll around in the camera view. One of them rolled around, survived for 4 months that way, rolling around in front of the camera. So that seems like an odd case, but almost always they're attached, and I can't say 100% of the time that they are.
Awesome. That was a great question. Rhea asks, are there sponge reefs within recreational dive limits?
Yeah, I wish. Not like the ones that you saw in the footage, those big extensive sponge reefs. Those are deeper than recreational diving depths, but you can see those same species shallower. So in the Puget Sound area, you can dive down to them, and actually they do start to form individual reef-like mounds called bioherms further north in British Columbia in Howe Sound. So yes, you can see those same species that form the sponge reefs when you're recreationally diving.
Great. And Mark asks, what is the time unit used for metabolism? Is it use of oxygen?
Oh, right. I would have to look at the specific graph. Sometimes we use different units. So please email me and I can look up for the particular one that I displayed. But I believe in the one I put in the talk, I was talking about micromoles of oxygen per liter pumped by the sponge. So it wasn't actually a unit of time. It was more a unit or a factor of how much water the sponge had processed.
Awesome. We actually got a question as part of our email, and it's a great question. Would you recommend a good book to read about sponges?
Ooh, would I? That's a good question. I don't have an immediate one, but I do have to say, if you want to look at some really neat classical literature about sponges, like from the research literature, there's this great research paper by Bitter, who was a sponge researcher back in the 1920s. And I think Bitter was just a fan of sponges. And so he wrote in the most poetic ways about sponges being a living screen between the used and the unused parts of the universe. And I don't know, it's probably the most whimsical writing I've ever read about sponges. But in the popular literature, I can't really think of anything off the top of my head.
Well, that sounds like a good book. Andy asks, why do you use microplastic beads? Is this only in the lab?
Yes, we use those in the lab for a couple reasons. One, because the sponges eat them, so we can use those to trace how much food is taken up by, or how much material the sponges took up. And importantly for that study, I spent a good part of my PhD graduate school days trying to figure out how to catch that sponge poop. That was actually like my white whale of my thesis, because I wanted to know what happened once the sponges ate particles. But if they digest material, I couldn't tell what the sponge poop looked like on the other end. So the microplastic beads were something that my PhD advisor, Sally Lees, suggested as something where we could feed them to the sponge and they wouldn't actually be digested, so we could trace them coming out the other end and recognize them on that other side.
Awesome. The next question, Deb is wondering what the flagella of the glass sponges are made of.
Oh, what are they made of? Well, they're part of each cell. So they're the same kinds of structures that make up other parts of cells. So there's a cell membrane on the outside, and on the inside, they have microtubules that help contribute to their stiffness and structure and let them wave back and forth as flagella do. So they're actually part of individual cells called the choanocytes.
Huh. And then someone's asking, what do live glass sponges feel like?
Oh, Good question. They feel a lot like dead glass sponges, which makes it a little tough when you're studying them. Smell is a good sniff test to tell, but actually they feel kind of like rice chips. They're very sharp. Some of them can be quite sharp if they have long glass spicules, and they'll be very crunchy because of that glass skeleton that makes up so much of their bodies. That said, I have felt some that feel very smooth, and so I'm like, oh, this one is nice, and I'll handle it without gloves. And then, have you ever handled fiberglass, and then afterwards you're very sorry you didn't be more careful? That can happen. So you'll get the little glass fibers from their skeletons in your skin. So don't pet a sponge without gloves. But yeah, that's what they'll feel like.
That's a great description. I can definitely imagine myself feeling that if I were to feel a sponge. Katie asks, how do species like the harp sponge generate their form?
Oh, how do they generate their form? That's such a great question, Katie. The true answer is we don't know. But thinking about it, why do they have the form that they do? Projecting up into the water column like they do gets the tips of each of those arms higher up into the water where currents might be stronger. And if you take a look at pictures of that harp sponge, you'll see that there are little bulbs at the top of those arms. Those are sperm packets that sit up there in those bulbs. So having the sperm packets up higher into the water column means that when they're released, those sperm can be broadcast further in faster moving currents, and so might have a better chance of reaching another harp sponge to help with reproduction and success.
Awesome. The harp sponge is actually one that got a big vote on that, on the red carpet anemone. So
that's my vote too.
Yeah, I think mine too. It's beautiful. Anyway, Tyler asks, do deep sponges offer habitat for other fauna like their euphotic congeners?
They absolutely do. This is one of my favorite parts of looking at sponge reefs and sponge communities, is just how much life lives in and among the sponges. So Keenan Gillis was a student at the University of Alberta, and now he's actually at Moss Landing Marine Labs, and he led a study during his undergrad where he studied all of the little animals that lived on top of the individual sponges, the little microscopic animals, and the sponges that lived on the dead skeletons, the animals that lived on those skeletons as well. So he found, I believe, over 100 different species just in a few sponges that he studied. And then also, when we've looked at a bigger view, we know that sponge grounds and sponge areas host many commercially fished species like rockfishes, including young rockfishes, like before they grow up to harvestable size. So they're really important as sort of homes for different animals to live in and around. And another fun one was in that sponge reef that we found down near the Channel Islands National Marine Sanctuary. That one was in the oxygen minimum zone of, and in that depth band, you find a lot of hagfishes. And so in any barrel-shaped sponge that we found, when we would look down with the ROV in the inside, there was always a hagfish nestled up inside of each barrel. It was sort of like your Oscar the Grouch moment. You'd peek in and you'd see this little hagfish head in there. It was great.
That's amazing. It's a great surprise. Bhavani asks, are the sponges able to get energy from the plastic beads? How are they impacted by small plastic particles in the sea?
Oh, I wish I had a better answer than we don't know yet, but we don't know yet. And to me, that is a really, really important next question to ask. So when I fed them microplastic beads, we did not ask or study whether any of that material was taken up. But clearly the sponges catch those plastic particles quite well. So it would be really interesting to know what they do with those particles. I know they poop them out, but whether they take up any of the material or whether it might be that they're actually have a significant sort of processing step of microplastics in the water would be really interesting to study. And that research just hasn't been done yet.
I guess there's always more research to do, right? Well, that's
Yes.
exciting. Katie asks, what cells cause sponges to contract, and are they similar to muscle cells and tissue?
Oh boy, Katie, that is a wonderful can of worms, can of sponges. I love that question. I don't think I can do it justice, but I can say that one of the really interesting things about sponges is, I know I put in my title, Understanding Ancient Animals, and where that comes from is that sponges are one of the first animal groups that diverged from the last common ancestor to all of the multicellular animals, and that means they're very, very different. They don't have to have a lot of the same traits that other animals do that were more recent divergences. Sponges do not have muscles, and many argue the prevailing view is that they don't have tissues either. So they're really a cellular level of organization. I won't get on my soapbox right now, but I do think that they have some tissue-like properties. But to answer your question, how do they contract? It would be the same mechanisms that would cause individual cells to contract. Things like the actin, which is found in cells and causes individual cells to contract, would happen and contract within the sponges as well. We don't know if there are any other specific mechanisms that cause the glass sponges to contract, but there are certain areas in non-glass sponges and shallower species and in the other groups of sponges that have a different level of organization. And those ones, they'll have sphincters, which are sort of these, holes that can tighten or open up more at different stages of their filter feeding system. And so they can kind of control the water flow through them that way and change the diameter of the canals that they pump water through. So we know a fair amount, but also there are a lot of details in there that make it really fun and complicated.
Awesome. That's a great question. Gets a lot of thoughts in the thinking about how all these things work.
Oh, it's so mind-bending. I love it.
Sarah asks, will ocean acidification affect deep sea communities and the sponges that live there?
Oh, such a good question, Sarah. Undoubtedly, yes. Ocean acidification will affect deep sea communities and already is affecting them, especially for animals that use calcium carbonate for their skeletons, like deep sea corals or snails. The acidification can affect the amount of calcification that they can do to build up and support their skeletons or their shells. And so having weaker shells or weaker skeletons means that they have to spend more energy to produce those hard structures, and also that they might have weaker skeletons or weaker shells that could be more subject to predators. For sponges, it's harder to know whether they would be harmed by acidification. Probably not for the silica spicules that they make for their skeletons, but we're not sure what might happen to their cells or their physiology. Those experiments also need to happen and are happening, where people can start to ask those questions.
Yeah. Serena asked, for the new species you haven't yet identified, how do you identify deep sea sponges? By their spicules?
Yes, exactly. So we're going to take a look at their spicules. So most of sponge taxonomy relies on those hard skeletons. So you will find out the types of spicules that are present, the sizes of them, and the particular combinations of spicules that make up a sponge, and oftentimes also the arrangement of them. So whether some spicules make a coat on the outer surface or whether they connect in tracks in different ways. And so that's the kind of work that we're doing to try to figure out what species we have in those sponge reefs.
Awesome. I want to recognize that it's 8:30, but there are a few more questions, and we've got some awesome questions that have been coming in. And thank you so much, Dr. Kahn, if you're okay with just a few more that we have. They've been so great so far.
This is so fun. Yes, I could talk about sponges forever, but yeah.
Awesome. Mary Jane asks, can asks, can sponges be used for chemical remediation or toxin removal in polluted waters?
Ooh, can they? It's a good question. I am not sure. I think that they are good indicators of clean waters for sure. So there are sponges in freshwater areas, and my PhD advisor, Sally Lees, she would always say that you could get a good idea of how clean a lake or a reservoir was if you would find freshwater sponges in them. And so we don't know what's the cause and what's the effect there, but definitely they like clean water, or else they are in clean water. But I'm not sure what their potential is for cleaning up pollutants.
Okay, and then Catherine asks, are sponges declining in population? And are there threatened or endangered species?
It's a good question, and it's a broad question. So in some places, sponges are at risk or are declining, and in other places, either we don't know or it doesn't seem like their populations are changing. So yeah, definitely protections have helped for the areas that have been at risk before. Those glass sponge reefs up in Canada, far north, now in Hecate Strait area, they're protected as marine protected areas. And that's because there used to be trawl fishing that would go through there, and the trawl fishermen would pull up nets full of sponges and say, oh, there's a lot of sponges down there. So now with no trawl fishing, those populations are more protected. So sometimes we know what the causes are that might be affecting declines in populations. Other times it's harder to say or to know. So part of the reason that that my collaborators studied the sponges in Norway was because there was a lot of oil and gas exploration happening there. And so they would drill, and they knew that the sponges were nearby, and they wanted to know what the effect was of the drilling or of what sorts of things might affect the sponges in the water. And one thing we see that really affects sponges is sediment. So resuspended sediments in the water can, regardless of the cause, whether it's caused naturally or whether we cause it by the activity that we're doing on the seafloor, those sediments can clog those filters or affect those filters and affect the amount of feeding that they can do.
This next question may be kind of related to the previous two you just answered, but maybe you have more to add to it. But are sponges being researched in relation to cleaning up pollution?
Oh, yeah, that's a good question. I am not sure. I don't personally know of any studies that are looking into that, but I can definitely look into that a little bit more. If whoever it was that asked that question wanted to email me, I can try to search around a little bit.
Okay, we just have, 3 more questions, so maybe we can get them all in. Paul asks, are you a diver yourself by any chance? And is that diving skill useful in this area of research?
Yes, I am a diver, and I love diving. It's not required for being a marine scientist. I do enjoy it though, and it is nice. I can't use it to study the deep sea sponges typically, although part of the perk of moving up to Canada during my PhD is that the sponges do come shallow enough there to study those glass sponges at diveable depths. But also now we are diving to study shallower sponges here in the Monterey Bay area. So yes, scuba diving is very useful for surveying and assessing both the density and the coverage of species that you see on the seafloor, and then also trying to study their pumping activity and their feeding and their effects.
Awesome. Megan asks, what lessons in biomimicry from sponges could we learn and apply for more sustainable societies?
Oh, I love that question. I often think about this too, Megan. So one of them, which I think that we humans have done a great job at, is exploring the medical potential of sponges. So Carol, that was news to me when you talked about that sponge that sponge extract that was tested against COVID-19. But definitely sponges have produced many secondary metabolites that have shown themselves to be useful for medical applications. And then the other one that I know of that people are studying for biomimicry is the spicules themselves, the skeletal elements. So when we make fiber optic cables, we humans have to use chemicals and high temperatures. When sponges do it, they do it with seawater. And those spicules are more flexible without breaking, and they still transmit light through them. And so it would be great if we could learn how they go about building those spicules. And finally, there's actually also some kind of cool, fun articles that study how those spicules are put together, almost like scaffoldings of a building or a skyscraper. And so sort of the architectural elements of how a sponge is built.
Oh, that sounds really cool. Okay, so I think we'll have, this is our last question. It's a really good question. It's from Barbara, and she asks, if they are cellular, it seems that movement would require their cell communication. How does that happen?
Oh, okay.
Or intercell communication. Sorry,
yes.
Would require intracell communication. How does that happen?
Cool question. Okay, couple things. First one is not all sponges are cellular. So the glass sponges, the ones that I focus on the most, they have, as a group, they have had their cells fused together into one common cell membrane. It's called a syncytium. So all the nuclei are housed inside underneath that common cell membrane. And so for them, communication is a little bit easier and a little bit different. So for them, they can actually create an action potential, which is the same thing that our nervous system does, basically a voltage that can help transmit quickly across tissues, or for us across nerves, to coordinate their bodies. And so in glass sponges, it's thought that because they are syncytial, they are able to kind of do things a little differently, respond as a whole animal and stop pumping. But in terms of the movement or contractions that we saw, we're not sure what communication is happening to cause that. And that's something that Sally Lees has been doing a lot of research on, and her students in the past. So things like what materials or what things in the water would cause sponges to contract. Sediments are one, but so are certain molecules, including signaling molecules like glutamate. And so those are things that can cause our nervous systems to fire, and they also can cause sponges to contract. For moving like we saw, rolling around on the seafloor, that was actually just currents that were rolling the sponges around. And so that one was a bit more passive. So yeah, it sort of depends on the type of movement, but that was such a great question. And if I didn't quite answer the particular type of movement that you meant, Barbara, I'd be happy to chat with you via email as well.
Gosh, well, Amanda, thank you so very much for this really fascinating evening. I feel like I got to travel to a whole new place and learn so much. And also really thank you for sharing it with all of us and the public, and it really will ensure that this new habitat is protected. So we really, really appreciate all the work that you're doing. It's so important. And I do want to thank everybody for joining us tonight. How cool is that, to learn about sponges on a Saturday night? So way to go. Great audience.
Thank you so much for inviting me. This was a blast. I will happily talk about sponges anytime, but definitely also getting to acknowledge and thank everything that the sanctuaries do to protect important sponge grounds. Like, thank you. It's just fantastic.
Great. Well, hope everyone has a great evening.
Thank you all so much.